Today. It is my pleasure to introduce today's speaker, Raina Plowright. Raina got her first degree as a veterinarian at the University of Sydney, then went on to get a master's in epidemiology and a PhD in ecology at UC Davis. Then she was the David H. Smith Conservation Postdoctoral Fellow at Penn State University, and moved on to be a research associate at Penn State and also at Griffith University in Australia. After that, she moved to her current position in the Department of Microbiology and Immunology at Montana State University, where she's now associate professor. And since then, or over this whole period, she's really built a thriving research program focused on pathogen spillover. And I would say, as someone who works in this field, that she's really emerged as a leader in this important interdisciplinary field. And I think, she is almost perhaps unique in her capacity to really realize the interdisciplinary and transdisciplinary nature of this work. She does, or I shall probably describe to you, her work spans disciplines from ecological field work to molecular virology, to bat immunology, to ecosystem scale processes. And I think it is really, perhaps the best implementation of the One Health idea, that all of our healths, human and animal and planetary, are interconnected, that I'm aware of by anybody, really. I should say I've been fortunate to be a friend and collaborator of Raina for the last number of years, so I've gotten a close-up view of a lot of this, and I know she has a lot of amazing stuff to show you. So I will shut up and let her get on with showing you the work.
Thank you, Jamie, for that very nice introduction. So you're looking at here, this is Pteropus medius. This is a reservoir host of henipavirus in Bangladesh, and I'll talk a little bit about them in a moment. Firstly, I'd love to acknowledge this amazing group of young researchers in my lab who get up to all sorts of adventures with our work. And also the broader collaboration of Bat1 Health, which is spans 11 institutions and 70 people, from PIs, technicians, postdocs, students, and much of the work that I'm showing today has been performed by these people. Our goal with Bat1 Health research or collaboration is to try to develop the scientific basis for spillover prevention. And we do that by thinking about known pathogens and new pathogens. So for known pathogens, we're looking at viruses in bats. So with the name Bat1 Health, you might have guessed, we focus on bat pathogens. So we're looking at bats in different regions of the world. We're looking at the patterns of viral excretion from these bats of known pathogens, mostly henipaviruses, Hendra and Nipah, but also now coronaviruses. We're then looking at the patterns of spillover in space and time. And then we're diving deeper into bat energetics, bat nutrition, immune function, movement, and behavior to try to understand why we see these patterns of viral excretion and spillover. We're looking at environmental drivers. We're combining everything together, all these different data sets together with different modeling approaches to try to develop predictions of when spillover occurs. And then we're looking at various countermeasures. As an aside, I'm not going to talk about this at all today, but as an aside, we are taking the viruses from the bats we sample in the field, and we're looking at the zoonotic potential of these various viruses through a whole range of different experiments in vivo, in vitro, and modeling. And so I won't be talking about that today. I'm going to start with these guys. So this is Pteropus electo having a drink of mango juice after we've just taken a blood sample from him and we're about to release him. So bats are really quite incredible, extraordinary creatures. They're the only mammals that fly. They're major, really important pollinators and seed dispersers in the systems where they exist. And they have these incredible life histories. So this is a Brand's bat, and it has been recorded at 41 years of age, and it weighs about 5 grams. So this ability to not go through senescence, to be able to fly, which needs high metabolic rates and should cause oxidative stress, is probably tied up with their ability to somehow host a range of pathogens. So many of the zoonoses that we think about, the Ebola virus, Marburg virus, the SARS-like coronaviruses, Hendra virus, Nipah virus, they have a bat origin. And the bats that do not make their bat host sick. And why that is still a mystery, but probably tied up with this flight and inability to withstand senescence. I should add, just before I go on, because there's so much talk about bats and diseases, is that I don't see bats as the bad guy. Bats really, they're like the canary. So these are really incredibly important species throughout their range for the ecological services they provide. And the fact we're seeing so many viruses spill over from them, I think, is an indication of something with our relationship changing with nature, as opposed to something that's I don't know, evil or wrong with bats. They are not the villains here. So let's step into thinking about pandemics from bats and what needs to happen for a pandemic to occur. Well, we have to have an infection, a pathogen in bats within their populations. And these pathogens are circulating and often have for millennia. Those pathogens have to be released from the host, usually through shedding. For example, coronavirus is shed in feces, henipavirus is in urine. There has to be a spillover process where the pathogen then contacts either a bridge host, intermediate host, or a human, and then there has to be spread amongst the human population. So for this infect, shed, spill, spread process, what happened for SARS-CoV-2, right, that we're all living and breathing right now? Hopefully not breathing, but certainly it's affecting all of our lives as we know it. Well, we know that the reservoir hosts for the SARS-like coronaviruses live in this region of Southeast Asia. They're horseshoe bats, and here's Wuhan. So what happened here? Did we have this infect, shed, spread, spill, spread process go on in Hubei province? Perhaps we had a bat taken out of the wild in Southeast Asia, was transported in a wildlife market to Hubei province, where it shed virus, which spilled over to a person or intermediate host, and then there was spread amongst humans. Or perhaps we had someone in Southeast Asia guano mining in a cave, was infected with the virus and then traveled to Hubei, or any combination, permutation or combination of all of those factors is possible. Unfortunately, we probably would never know how that original spillover event occurred. And if that's the case, how are we going to predict the next pandemic? Well, if we think about coronaviruses, they are everywhere. More than 3,000 coronavirus variants in bats. There are coronaviruses in bats in every continent we find bats. So that's everywhere except Antarctica. And in most bat families, coronaviruses have been detected. And there's probably around 200 at least SARS-like coronaviruses that could be zoonotic. And if we think about the coronaviruses that have caused major outbreaks, we've had 3 cause major outbreaks in the last 20 years. SARS-1 went bats to civets to people, Middle East respiratory syndrome, bats to camels a long time ago, and then camels to people, and then SARS-2, we don't know. But essentially, all of these processes may have originated with a single spillover event from a bat to an intermediate host, leading then to propagation to people. So we may have n equals 3. And with n equals 3, we just don't have the data to build up the patterns to really understand the processes. So what our group is doing is we're going into systems where there is spillover, and pathogen spillover is a common occurrence. So we're working in places like Bangladesh with Nipah virus and Hendra virus in Australia, where here you can see here we have space, latitude, and time, and every dot here is a cluster of Nipah virus events. And here we have space and time with Australia, and we can see every dot is a spillover event from bats to horses for Hendra. And so now we're in systems where we have regular spillover, so we can start to study the process of spillover to start to understand what is driving this process. So again, Tropus medius from Bangladesh. Nipah virus is one of the WHO top 10 priority pathogens, along with Hendra virus. And we're worried about Nipah spilling over in a place like this, where we have high density human populations and poor public health infrastructure. And with Nipah virus, we have a very distinct form of spillover where people get Nipah virus from drinking date palm sap. People put a gash in the tree, the palm sap drips down, and during the night, while the sap is dripping down, the bats come and actually lick the sap off the trees. And if you watch carefully, this bat's going to turn upside down and pee right in that collection pot. And then you can trace Nipah virus outbreaks. This is a paper by Luby et al. And this is the root of the date palm sap cellar. And the red dots are outbreaks or people who are infected with Nipah virus. So it's a beautiful epidemiological study there of the spillover of Nipah virus from bats in Bangladesh. In Australia, we don't have date palm sap consumption, and I think if we did, we'd probably get Hendra virus spillover through date palm sap consumption. In Australia, Hendra virus, in the same genus as Nipah, is endemic in the 4 species of fruit bats, Pteropus species fruit bats, or I'll call them flying foxes. And Hendra virus is shed in the urine, and when this happens, when a bat is feeding in a paddock or a horse is grazing, they can contaminate the grass with Hendra, and then a horse grazing that grass can then become infected. And horses are very susceptible. There's about an 80% fatality rate in horses, and there's been 61 spillover events from bats to one or a group of horses. And then people get Hendra by having contact with sick horses. Thankfully, only 7 cases of Hendra in people, 4 are fatal, but that's still a 57% fatality rate of Hendra virus, and a terrible tragedy for the people involved. Also, it's really changed the veterinary industry in Australia now that any sick horse could essentially have a biosecurity level 4 zoonotic pathogen. So I started working in this system for my PhD. And this is just a little bit of background here. For my PhD, I worked on little red flying foxes, and I caught bats from the same population over time, so repeatedly sampling the same population over time to look at these patterns of viral excretion. And we didn't always work on boats. We just did this because this part of the Northern Territory, there were a lot of crocodiles on the banks of the river where the bats were roosting, and we got a little bit wary of the crocodiles, and we thought being in a boat was safer until they caught a big crocodile at the boat ramp. But it was a very effective way to catch bats. And these are, as all of the species in Australia are, these are nomadic nectivorous animals. So they form huge aggregations of animals, and they continuously move across the landscape looking for big bangs of flowering in native forests. So these circles here show populations moving around, disappearing and appearing across the landscape through time. So very dynamic in space and time, very ephemeral populations. And during that period, I had a serendipitous finding during my PhD where we happened to sample bats during a large hurricane, which we call cyclones in Australia, and there was a food shortage and the bats were starving. We had a starvation event, and we found during that period very high levels of Hendra virus, higher than at any other point in time. And that really led to what we're doing today. So when I was studying those bats in the Northern Territory was in this period here, and to be honest, I don't think anyone really cared about Hendra virus or really had heard about Hendra virus, and certainly didn't care much about bats and viruses in general. Hendra had been this sporadic, one horse in a paddock type of disease, except for the first outbreak, which was quite spectacular. But around the time I finished my PhD around here, we started to see Hendra every year. And then we started to see big clusters of Hendra spillover. So what we now see is we see a cluster of many events. In 2011, 17 events. And then we'll see no virus for another year. And then we'll see virus, another cluster of viral events here. And I just wanted to point out, in the subtropics where we work, almost all Hendra virus spillover happens in winter. So around this time, I started working with Peggy Eby, Dr. Peggy Eby, who's a bat behavioral ecologist who'd been collecting long-term data on bat distribution and behavior on the east coast of Australia. And she'd said she'd been present at some of the infected premises where the horses had died during these outbreaks. And she'd noticed the bats in that area were exhibiting starvation avoidance behaviors, meaning those bats were behaving like there was no food. They were eating things that bats don't usually eat. They were eating various food types that just don't have very much nutrition. And they had very high secondary metabolite content. And so Peggy and I we put this together. Maybe there's an interaction. We're seeing high henipavirus. Maybe we see high henipavirus when the bats are starving. We're certainly seeing these behaviors during spillover events of bats starving. So we started to think, well, what's driving these clusters of spillover? Is it something to do with the reservoir host distribution? Is it something to do with the virus within the bats? And so we wrote some grants. We were really fortunate to be funded by a National Science Foundation coupled natural human systems grant, and then more recently DARPA funded to look at the patterns of viral excretion, Hendra excretion in bats over space and time, to dig deep into these spillover events. So we've been able to send a team to every spillover event in the subtropics during our project, where as soon as we hear about a horse that's come down with Hendra virus, we have a team on the ground investigating what are the circumstances surrounding that spillover event. And then to also be able to look at the condition of the bats during these times of peak shedding. And of course, the various drivers that might be driving these peaks. Really critically is that we're able to assemble a long-term, so 16 long-term data sets, so that span over 25 years on the bat behavior and distribution in Australia. We've been able to replicate some of that in Bangladesh, Madagascar, and Ghana, but with very little time, I'm going to focus on Australia today. So we have field teams, we sample the same populations again and again, and what we do is some of you have done this. I had some good meetings before this talk. We have large mist nets. The bats fly into the nets as they're returning from feeding. We also collect urine and feces from bats under roosts with big tarpaulins. It's not usually this dramatic, but we do this, and we resample the same roost every month. When we catch individual bats, we are able to do a suite of different studies on the condition, immune status, and so on of those bats. This is them going into the pillowcase, and then they get an anesthetic and blood sample, and many different samples are taken from those poor bats, and then they're released back into the population where we caught them. So I'm going to jump now to some of the results that we're seeing. So what we do see, this is prevalence. So this is the proportion, actually it's the proportion of this is under roost work, so proportion of urine pools that are positive for Hendra virus. And this is over time. And what we see is we certainly see these peaks in shedding and prevalence, and they tend to align with winter. This is winter here, when food is short. And we see higher peaks during periods of food shortages. So this was actually during a severe food shortage and a cluster of Hendra virus outbreaks in horses here. And this is actually using, this is modeled from our field data. This is the area, using an area under the curve approach with GAM models, and then trying to actually model Hendra virus pathogen pressure, like the amount of virus that's actually available to those horses over time. And then really interestingly is what we've found is not just the peaks that matter. So it's not just how many bats are infected or the proportion of bats that are infected at any time, it's how much virus they're actually shedding. And so what we've found, some new work that Tamika Lunn is preparing, is that when we look at the viral loads, so CT value being lower means more virus. When we just look at, we use CT30 as a cutoff, we see more distinct winter pulses of virus. This is data aggregated over our field sites. And when we have spillover events, we have CT values, we have viral loads, very high viral loads in the bats when we see spillover events in horses. So the intensity of infection in those bats actually really does matter. I should say, and I'm not presenting this data today, but what you also find when the bats are shedding in a pulse like this in 2017, we're finding they're shedding many different pathogens. And Alison Peel in Australia led a paper that was out last year from our group. She led a paper showing many different, so 9 different paramyxoviruses being shed with hendrovirus at the same time. So it seems like some underlying process is really driving the shedding. So we looked at bat distribution, and much of this data was collected actually by Peggy EEB, my collaborator in Australia, over time. And excuse the graphs, because this is really a plot that's really in progress right now for a current publication. So what we've found is that this is the number of flying fox roosts in our study area, which is a small area. It's an area that encompasses southeast Queensland and northern New South Wales of Australia. That subtropical area where just about every spillover event in the subtropics has happened. What we found in that area is that at the beginning of this period, there was a stable number of roosts, about 100 roosts. We'd see occasionally there'd be a fission of roosts during a food shortage, but they'll quickly come back to their stable large aggregations. But then we saw a rapid increase in the number of bat roosts, a tripling actually of the bat roosts. And we noticed that those increases are stepped. They happen in a stepped way. And each of those steps aligns with a period of food shortage. And we measure food shortage in bats by 2 variables, the reproductive success that year. And so we find that after a food shortage, the reproductive success is lower. So the percentage of females with young is low. And we look at the surge of bats, like these little guys, juvenile bats usually, coming into rehabilitation hospitals, wildlife hospitals. And we see a surge of bats coming into these wildlife hospitals during food shortages. And you'll notice then the food shortages we can track down to show also result in this increased number of roosts. And what we also have shown is that these food shortages are most of the time preceded by a high oceanic Niño index. This is an index of basically the temperature in the middle of the Pacific Ocean. So it's related to the La Nina, El Nino cycles. But when we have a high El Nino year with a high ONI, we always have a food shortage for bats in the following year. We also have food shortages for bats that are independent of ONI. But every time ONI crosses 0.8, we get a food shortage for bats. So what we've understood is that increase in the number of roosts is not an increase in the number of bats. In fact, we've got population estimates showing, if anything, the number of bats is stable or decreasing. What we think is happening is it's a fissioning process. So historically, what we would see was climate oscillations. So you would see a high ONI, we'd see an acute food shortage, and this might last for a few weeks. And these big nomadic populations of bats would quickly fission into these small populations that go probably traditionally into rainforests, but now it'll go into urban areas and agricultural areas. They'll find food that we provide, like fig trees in horse paddocks or fruit production trees or trees we plant in the city streets. But historically, they would then quickly go back to nomadism, and this might last 2 or 3 weeks. And so it's not even captured in the data we have. What we're seeing now is that these nomadic populations are fissioning into small populations, and they're not going back. They're not going back to nomadic. So we're just seeing this exponential increase in the number of populations. In another study, which I'm not presenting today, we did an analysis of spillover events, and we found these new populations, these new fission populations, are almost always responsible for the spillover to horses. And this fissioning process of the number of roosts increasing in these steps with food shortages is happening across the east coast of Australia. So this is not just happening within our area. And the reason we're not seeing spillover events here is because the species of bat is not a good reservoir host of henipavirus. So we've looked then at, well, where are these new populations of bats moving to? And what we've found is that over our period of study, the bats have become more and more urban. So they've gone from feeding in native forests to essentially feeding in agricultural and urban areas. And these are just a few data visualizations of this process showing this is the extent of feeding areas classified as agricultural, urban, increasing over time. And then this here is really straight out of the modeling that we're doing right now. Every dot that you can see is a flying fox roost across our study period. And the blue dots are roosts that are in urban areas. The red dots are in this sort of mixed use area of agriculture, essentially, where there's human dominated activities and animals. And this is forested areas. And then the black dots are the spillover events. And so you can see that the bats, they're moving into urban areas, they're moving into agricultural areas with this fissioning process, but the spillover events are happening in the agricultural mixed use areas, which absolutely makes sense because the intermediate host for this pathogen is a horse. And so we're not going to have horses in the middle of a city. We're going to have horses actually in agricultural areas. Another clue that we have here is that we've put GPS transmitters on bats looking at their feeding behavior. This is a Tropiselecto bat, so this is the most competent host for henipavirus we have in Australia and really defines the distribution of the virus. He's flying off and look at his movements and where he feeds over the next 10 days with blue GPS transmitter. What we found is that during a food shortage, these bats with the GPS transmitters were feeding in horse paddocks on food that we provide. So fruit trees, for example, in horse paddocks. But when nectar is available, the bats would amass back into big aggregations and then fly to the nectar, and they'll be feeding in native forests away from horses. So during a food shortage, 50%, more than 50% of the bats' feeding stops were in horse paddocks. But when nectar was available, there was virtually no feeding stops in horse paddocks. So behavior is a really important part of this dynamic. It's not all just the virus being, it's not all about viral shedding. It's about that contact process as well. So I wanted to go back to this diagram as well. As you can see, very much an in progress diagram here. So these food shortages from bats caused by these climatic oscillations, they have probably happened forever, for the entire time that bats have been on the Australian continent. So why is it then that we didn't start to see a change in the ecology of bats until around this period here? Around 2003 was when we think things really changed. So I want you to hold on to that for a second. I also want to point out that, yes, we had climate oscillations and food shortages throughout this period, but we did not have henipavirus spillover. We only started seeing henipavirus spillover in this period really rapidly starting around 2006, but certainly around 2004, we started to see some events. And these circles here are spillover events. And so what we think is happening is that when we have a fission bat population that's now living amongst horses, and then we get a food shortage, they're more likely, they're shedding virus, but they're shedding it where the horses are. And so they're more likely to cause a spillover. Whereas if they're shedding virus here in a native forest or somewhere where they're away from horses, it doesn't really matter because we haven't got the right host available for spillover. And so what we've seen is this pattern again and again of high ONI food shortage. Here, high ONI food shortage, and then high ONI food shortage henipavirus that next winter, high ONI food shortage henipavirus the next winter, and so on and so on. Why now? We've done some work now looking at feeding habitat across this whole time period, which I'm not going to show for time, but one thing that we know, and this is work from Peggy EEB, is that these nectivorous bats have a lot of habitat available in summer. In fact, it's all mostly in montane, well-protected areas. Many species of trees produce nectar in summer. But in winter, there's only a few species of trees that provide reliable nectar. And those few species of trees, they live in these they live in coastal lowland habitats that are very important for agriculture because their soil is very rich. And so they're almost completely cleared. And we've looked at the loss of winter feeding habitat of the bats over that study period I showed you there, and it's just been a consistent decline in winter habitat. In some areas we've lost 96% of what was originally there. And so what we think is happening is the bats, essentially they get to winter and there's a food bottleneck, and that food bottleneck, then if that's superimposed on with a food shortage, we see then these rapid changes in their ecology. So we sort of have a sense of what might be going on here, but thank you to reviewer number 1, who suggested we actually really need to put all those 16 data sets over 24 years into a model to rule all models. So we decided to, we've been working with a statistician, Dr. Andy Hoig at Montana State University, to try to come up with a modeling framework for the system. And so we've developed this Bayesian network model where we've now got data on all the climate isolations over time, all the food shortages over time from those incredible data sets that EEB had collected. And the model actually shows, yes, ONI predicts food shortage, and food shortage predicts population fragmentation into new winter ranges overlapping with horses. And food shortage predicts spillover. And in fact, our models show that at a population level, at the roost level, like a single roost of flying foxes, in a high ONI year post-food shortage, there's a 15% chance per roost of a spillover event. Okay, so now I'm going to talk a little bit about prevention. So I showed you our predictors of spillover, and we did some machine learning modeling earlier before we did the Bayesian network analysis, and we found we could pretty much predict these food shortages. And we had a really good sense of every food shortage followed by henipavirus outbreaks. And then in 2019, we had a high ONI. We then had a huge surge of bats coming into rehabilitation. And we had the lowest reproductive success we'd ever seen in bats. And this is from a survey that Peggy does up the east coast of Australia every year and has done so for 25 years. So we predicted then 2020 was going to be a terrible year. On top of that, some of the large areas of big flowering habitat for bats in winter were burnt in the Australian bushfires that past summer. So we thought, well, we're going to have spillover. And we'd predict the spillover before, we'd been right. And so we had an alert go out through the chief veterinary officers in Australia to vaccinate horses. And I just let you know there is a vaccine for horses. It's very effective, but it's not used. There's been very low uptake of the vaccine for many different reasons, including anti-vaccination narratives, similar to the MMR vaccination. So in this case, we said people should vaccinate their horses in response to this alert. We actually just got the data back from the vaccine company. And even though we had a paper go out in the veterinary journals, we had alerts go all over the equine world, we did not see an uptake in vaccination. And so I just put that out there that, again, social science, we really need that. We need to think about how we're going to implement these sort of interventions. And we actually have a social science project within our coupled natural human systems work, looking at narratives to encourage people to uptake vaccinations. Anyhow, what happened in 2020 is that we had an early spillover event. So we had an event that actually happened in May 2020. And we thought, that's very early. That's right at the beginning of the austral winter. We're going to have a terrible spillover season. We had big teams ready to get onto every property with spillover. We were all set to go. And then nothing. No more spillover events. And we thought, well, let's figure out what's going on. So thankfully, we had a lot of people in the field because we were sampling animals. And our field teams discovered through huge amount of research and networking that there was a major blossoming event happening in Queensland within our study area. And this is very typical for a flying fox roost now that they're in these urban, peri-urban areas. This was the flying fox roost before there was a flowering blossom event. And then by July, there were 200,000 bats here. Actually, 75% of one of the species, Pteropus poliocephalus, was in this roost, feeding on the Queensland blue gums. And we went back through data, and Peggy had already, with her long-term knowledge and intuition in the system, had been thinking there seemed to be something about flowering going on with spillover. And she'd already noticed that when there was a big spotted gum event, these happen every 4 to 7 years on the South Coast, and they attract hundreds of thousands of bats from like 800 kilometers. Bats will come to these events. She had noticed that every time we had spotted gum that we didn't see spillover. And what we now understand is that actually we'd never had a big pulse of winter flowering during a year where we had also just had a food shortage. So that what we saw in 2020 was the first time we've had a predicted conditions for henipavirus, for post-food shortage for henipavirus, and had a flowering event. So what we did is we decided we put this into the model, and this took a huge amount of work that Peggy did, where she went through all of the historic data on flowering across our study area and beyond, looking at winter flowering events. And here we have spillover and flowering. Spillovers are in red, and that is a mistake there, I think, from my messing around in Excel. But what we now understand is that there's actually never a period where we have a spillover event. These are replicates of Australian winters. There's never been a winter where we have a spillover event at the same time as flowering, as winter flowering. And so what we did is we took those data and we put them into the Bayesian network model again. So we incorporated flowering, and what we found is that as before, climatic oscillation predicted food shortage, which then was related to population fragmentation into agricultural and urban areas. Food shortages were predictive of spillover, but flowering pulses stopped spillover. And so, like I said before, here, if we had a food shortage, we had per roost site a 15% chance of a spillover. But if we had a flowering pulse that winter, we essentially had a 0 chance of spillover. And so this really begs the question then about how perhaps if we had enough flowering habitat, we had the diversity and abundance of habitat that flowered in winter. If there was flowering available for bats every winter, then maybe we could actually stop spillover. And we feel that perhaps that 2020 event where everything lined up to have spillover in horses, and then the flowering essentially stopped. What we believe is it stopped that spillover process. Perhaps what we could do is we could see a reversal of the system where nomadic populations or fission populations can become nomadic again, can revert to feeding on nectar and blossom rather than fruit in agricultural areas, also regaining that pollination service that bats provide. So we're actually working on a program, we're calling it Ecological Countermeasures for Public Health, where we're trying to replant these particular trees. So in conclusion, it's by understanding that root cause of spillover, which we think is this loss of winter habitat leading to winter food bottlenecks, superimposed on these natural food shortages, fission events into agricultural areas where bats now feed amongst horses. And then there's viral process on top of that when bats are stressed and nutritionally stressed, shedding more virus in areas where horses are. Since we're getting to the point where we understand this entire process, now we can start to think about how could we stop it at its very root cause. So rather than vaccinating every horse for henipavirus, which seems to be difficult because of human behavioral limitations and economic limitations, perhaps we could actually fix this problem right at its source. It also shows how long-term studies of reservoir hosts and pathogens are really essential. So interestingly, I think this is really the only system where we have long-term data on the structure of a reservoir host through time, showing it rapidly changing in response to environmental change. And we've actually gone to Bangladesh now and looked at the structure of bat populations and deforestation rates in Bangladesh. And we see really Bangladesh is probably where Australia is going. It's essentially every bat population lives within a human community, and deforestation happened decades ago, actually more like 100 years ago. And it is essentially a human-dominated environment. But also shows us maybe we could address pandemics if we really understood what drove them. And I'll just finish by showing a bat being released. Thank you.
Very nice, Raina. Thank you. That was brilliant. And you were so disciplined with your time that we have plenty of time for questions now. And I see they're beginning to fill up the Q&A on Zoom. So since others can't see that, I will read questions to you. I think you can probably read them as well, but others can't see them. And then we will let you respond. So the first came in from Benny Boremans. He says, going back to the broader context of predicting spillover, I would love to know your thoughts on ongoing monitoring to assess variation in spillover risk. Which strategy do you think should be prioritized? Monitoring for pathogens by antigen or antibody testing in humans, monitoring key animal sources, or something completely different like environmental conditions that might be linked to reservoir behavior and shedding?
Wow. Hi, Benny. Nice to hear from you. Big question there. Like, how do we stop this all happening? I think what the Australian system is showing us that you need some baseline data to understand how these systems are changing and responding to the pressures, particularly the recent environmental pressures on these systems. So how do we get these baseline data so that we can see when there are perturbations and then understand what's driving those perturbations? That's really tough in many places in the world. And I look at the places where I think spillover is most likely. We will have spillover in Australia, but it's probably not going to become a pandemic because we'll detect it and contain it. But if we have a spillover in Central Africa or parts of Southeast Asia, that we may not be so lucky. So that's where we really need that baseline data in those high-risk systems. But we're working in places there where there's very little capacity. And some of the countries where the horseshoe bats are endemic, there isn't even a zoologist, let alone a bat ecologist. And so how can we develop those sort of data sets? It needs a lot of investment. Then I think that understanding the ecology of these systems, we know virtually nothing about the ecology of most of these systems. We're doing a review of bat coronaviruses right now, and we've looked at all of the longitudinal and spatial data in these systems, and there is one study that might be called longitudinal, but even so, it actually pulled samples over space. So there isn't actually a spatiotemporal study of coronavirus shedding in bats. So what are the stresses that are driving the virus dynamics in bats and driving the virus out of bats and into humans? And then, of course, there's the human interface and the intermediate host interface, which is really, really difficult work. And that needs a high level of specialized work in the social sciences. And that needs to be integral to all of these different programs. So I think in general, it needs a huge amount of transdisciplinary excellence at a level that we don't generally perform and a level that's not generally funded. And so we're going to need like serious funding and serious incentives for people to work together across all these different spatial scales and disciplines.
Great, thank you. Next, I see a note from Dan Blumstein that he has a question, and I see something typed in later, but I'm going to allow Dan to talk in case he wants to voice that and another question together. Oops, I just unmuted the wrong person. I will find Dan now.
Am I unmuted?
Oh, there you are. Okay.
Okay. Fabulous talk. Really super interesting. The social science aspect of the vaccine non-uptake is fascinating as well. But I'm a behavioral ecologist. I'm interested in how much variation and spillover might be driven by the pathogens changing the bat behavior In other words, is there any evidence that it makes sick bats more likely to move around under food stress?
Oh, that's interesting. We know, but the kinds of studies you'd need to do to address that question would be really difficult. So right now, the paradigm in the field is that bats do not get sick from these viruses. But sublethal effects of these viruses are very hard to study. We really need to be tracking the same bats over time, and it's very hard to catch the same bat twice. So those studies haven't been done. Would the bats move around in a different way? Well, yes. What's causing what? Okay, that's hard. I would say a more parsimonious explanation is that there is a shortage of food. The bats are starving. They don't have enough energy to move. And so instead of flying 50, 80 kilometers to find nectar, which is probably not going to be there, they move 1 kilometer to eat some crap food that's in the next paddock. And that happens to be in the environment where horses are. So I think it's essentially this is an energetic problem that's driving the behavior of bats, just not being able to move. And when we catch bats that are in a pulse of nectar, it's completely different behavior. So these bats are like, I don't know if you've ever watched a child drink a bottle of soda and then just let the next 10 minutes play out. And they just basically go, and they start bouncing off the ceiling and running around, and it lasts for a little while and then they collapse. And it's horrific, it really. Well, the bats are almost like kids on soda. When they have nectar, they have so much sugar. They're flying 50 kilometers, 80 kilometers over here. They're bouncing across the landscape. There are actually records of bats having to fly further just to burn off some of the sugar. They've got such a high amount of sugar. So really different behavior. And it's behavior that's taking them away from horses, away from that recipient host. So yeah, I'd say that's probably more likely than the virus driving the host. But we wouldn't be able to address that question at the moment.
Thank you.
Great. Next question is from Morgan Tingley, who says, reforestation is an obvious solution, but it's long-term, not short-term. Are there other short-term solutions? Can fruit trees be fenced off from horses to separate horses from bats? Can supplemental food be provided in safe areas to concentrate bats in non-spillover areas?
Yeah, very much so. And it would take about 20 years for a tree to produce enough nectar to be able to feed bats. My first postdoc in my lab said to me when we were discussing this, he said, Raina, we should set up mango juice stands outside bat colonies and feed the bats. And I remember thinking, I've got to get him in the field. Once he sees the bats, he'll be okay. But, I have to laugh now because now we're thinking, how do we really test these hypotheses? And what we need to do is those kind of field experiments where we set up mango juice stands. I don't know if we could actually get bats to take some kind of supplemental food, but during the really severe food shortage at the end of 2019, people were placing like rings of fruit in their backyards with apples and bananas and things on them to feed the bats because they were really in dire straits. So I think it would be possible to do that. Really complicated to do that as a field experiment. In terms of the horse, yes, the most simple thing that we can do is keep horses away from trees where bats feed. And so fencing off horses, at least at nighttime, because the virus probably only survives for a few hours on the ground. So if you just keep them out of the trees at nighttime and then let them graze as normal in the daytime, then we'll probably stop horses getting exposed. And those kind of, that sort of information is being really pushed on the horse community, is don't put your water source under a tree. Keep bats and their trees and horses separate, and then vaccinate. And vaccination seems so simple, but it's been actually, sometimes that human behavior stuff seems more complex than the biology we're trying to understand.
So that perfectly sets up the next question from Pat Arena. Who says, can you provide more information about the anti-vax in the equine community? Is it because of a concentrated effort among anti-vaxxers, or is there general ignorance in the community?
It's been a social media driven effort. And so there are Facebook sites and so on that are related to anti-vaccination campaigns. And so my original training was as a veterinarian, and I worked with horses, and I know, and anyone who's worked with horses, that if you touch a horse and it can die, you stick a needle in a horse and it's certain to die. but anything can happen to horses for some reason. They seem so susceptible. And so you go to the Facebook site, and basically anything that's ever happened to a horse, like lameness, broken leg, sudden death, it's attributed to the vaccine. And which you can imagine, because if you're vaccinating a lot of horses and a lot of horses are dying, you're going to have sometimes horses are dying right within 2 weeks of a vaccination. And of course, the brain naturally wants to make a causal association there. There are economic reasons. Right now, there's a tight certification of the vaccine, so a veterinarian has to administer it, and that's expensive to have a vet come to the property. And then there are also some trade regulations with certain countries, some countries only importing horses that are vaccinated, and some countries only importing horses that are not vaccinated. And cost for a huge, like say, racing stable with or a farm with 10,000 horses, it's going to cost a huge amount of money. So many reasons. And the way that we're addressing this is we have a really incredible social scientist on our team. Her name's Liz Shanahan, and she's developed something called narrative policy framework. And what it is trying to deliver science within a story, the framework of a story. So where you have heroes and you have villains and you have actions going on. And she's found that if you place the science with the person that you're delivering the science to as the hero of the story. So you vaccinate your horse and you're protecting your horse and your kids and your property from this terrible event. You're like this hero. That sort of narrative is very appealing and it makes people more likely to change people's behavior. So right now we're doing these big surveys to horse owners with different narratives and seeing which ones work better for vaccine uptake. But it's a long way to go. I think it's really complicated.
Amazing. Another question from Nitika Sharma. Thank you for sharing the incredible study that weaves together so beautifully. You mentioned the presence of fission groups for prolonged durations instead of remaining to big nomadic social groups causes spillovers. I understand that this is due to their altered behavior where they hang out near horses more. I was wondering if you speculate an additional role of limited allogrooming in smaller social groups as compared to larger social groups. I'm thinking of the context of a few studies indicating popular, highly interacting individuals having better immunity in social groups.
Oh, that's interesting. Yeah, okay. Well, I hadn't actually thought about it in that respect, so thanks for that. It's in infectious disease ecology, we always think about things as usually density dependent, right? As you throw a whole bunch of people into a bar for a long period of time and you're more likely to get COVID. Well, the same with bats. You throw a whole bunch of bats into a tree, maybe they're more likely to transmit the henipavirus. You're likely to have a higher proportion of bats infected. And so we should think then we get these huge aggregations of bats around these nomadic feeding sources, flowers, we should have huge amount of henipavirus. That's not what we're seeing, although I have to say our data on that are still very much in progress. But what we see is we see these high shedding events when the bats are in those very small populations in the middle of winter, in those fission situations where they're energetically stressed and they're just roosting very close to some sort of food source. We've actually watched them in food shortages split into smaller and smaller groups to the point where you can just have a few individual bats just roosting right near a tree that they're feeding in because they're just so energetically stressed. So we think that's probably driving the problem. But I have to think about the allogrooming. Thanks for that.
Great. One more from the Q&A from Joey Curdy. You mentioned that in one species of pteropid bats, there's approximately 75% of the whole population converging on a single eucalyptus flowering event. Do we know well how so many bats can be drawn to a single location? What is the key for this? Are they smelling over a long distance? Furthermore, are there any current efforts to establish more populations of these flowering eucalypts?
I wish that's like the biggest question, I think, in this nomadic ecology. How do they know, and how are they sensing this from such a long way away? And the theory is that this is a reason why there are very big aggregations, why these bats have evolved to be in very, very large groups, is that they are able to more easily share information. And so as bats come into different roosts and sense what other bats are feeding on, they get a sense of what sort of resources are around. But we really don't know. We really don't know how they're detecting these flowering events over such long distances. There was a second part of that was the so yes, so we're at the moment, so we're working in two scales with the replanting. We're working from the bottom up grassroots scale and then from the top down policy scale. So from the grassroots scale, we're working in community groups and just letting people know what species of trees to plant, because there are huge replanting programs in Australia that are funded by the government. So it's really a matter of just using the right trees in the right places. And often a lot of the narrative framing around that is around koalas and nectivorous birds, because it's a whole nother story, but Australians tend not to feel very positively towards bats, because there's been such an inundation of bats into urban areas and they've been so disruptive. They're smelly, they're loud, and so there's a lot of like anger, a lot of very strong feelings, negative feelings about bats. So, but no one hates koalas. Personally, I think bats are cuter, but it's much easier to get someone to plant a tree for a koala than a bat. So, well, whatever, they both use the same trees. So that's the bottom up process. And then the policy process, we're started on the really trying to understand how these mechanisms work, how this would work, how much is there. So really doing the science well, and then communicating that science through a bunch of publications, and then working with policymakers, step-by-step process. And we're working with someone, we have a team member who is a policy expert working with us on that. And that wasn't our plan, actually. We were going to go bottom up, but with the pandemic and having a pandemic of a bat origin disease, we think there are probably more opportunities now to work more at the higher level with this.
Great, thank you. I think we have one more minute. I'm going to abuse my host prerogative to ask you one question, just see how far I can entice you into generalizing these ideas. So like you mentioned the extension into Bangladesh, which I think is fascinating, but if we think about coronaviruses, if we think about filoviruses, the story of Ebola, the 2013 initial spillover event in some lone tree in a village, how far do you think these ideas of anthropogenic landscape change, stressing out bats, basically giving rise to higher spillover risk, how far can we go there?
Well, everything's speculation because there's zero data, so I'll speculate. Many of those areas that are undergoing spillover events are having they're in rapidly transforming landscapes. So especially West Africa, Central Africa, where we're seeing huge expansion of palm plantations, for example, in Southeast Asia as well, expansion of agriculture. And we have bat species that have similar ecologies. So nomadic bats who depend on fruits and nectar from forests. And so I can just extend my imagination to think that there is probably rapid loss of habitat, rapidly changing availability of resources at different times of the year. And that's probably changing the distribution and behavior of the bats. And it's probably changing also the stresses on bats, their immune function and their ability to hold these diseases and to be excreting these pathogens. The problem is there's no data and there's just no long-term studies. There's not even the people to do the studies, let alone the money to do the studies. And it's a huge hole, it's a huge gap. But I think it's just critical. It's just critical we're doing these. We should be doing these, especially in Southeast Asia right now with coronaviruses, so we can know what's going to happen with the next coronavirus.
Indeed. And I'll just opine that I think your work is visionary in putting forward an actual mechanism for how the landscape change could be leading to the observed pattern of enhanced spillover risk in those areas. Okay, thank you very much, Raina. Thank you everybody for joining us today for Raina's seminar. And with that, I send you off to your afternoons. Thanks very much.
Thank you. Thanks, Jamie. Thanks, everyone.
Thanks, Raina. That's great.